na, humanda sabay-sabay akyat. Hawak kamay tayo'y aangat, lilipad, ating abutin ang pangarap ni Juan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan kung lahat magtutulungan. Tara na, sama-sama itaguyod ang siyensya. Maayos na bukas para sa Pilipinas. Ating abutin ang pangarap ni Juan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan kung lahat magtutulungan. Tara na, sama-sama itaguyod ang siyensya. Maayos na bukas para sa Pilipinas. Maayos na bukas para sa Pilipinas. Maayos na bukas ating iparanas sa bayang Pilipinas. Ating abutin ang pangarap ni Juan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan.
Tumitilaok na ang manok, hudyat na ng pagpasok. Paglilingkod na walang kapalit sa bayan ng aming hatid. Tara na, kaibigan, huwag kang magpaiwan. Gamitin ang dunong bansa'y susulong. Ating abutin ang pangarap ni Juan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan kung lahat magtutulungan. Tara na, sama-sama itaguyod ang siyensya. Maayos na bukas para sa Pilipinas.
Anumang hamon ay haharapin, mahirap man ay kakayanin.
Sa pinagsamang lakas at galing, tagumpay ay mararating.
Tara na, kaibigan, huwag kang magpaiwan.
Recording in progress.
Gamitin ang dunong bansa'y susulong. Ating abutin ang pangarap ni Juan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan kung lahat magtutulungan. Tara na, sama-sama itaguyod ang siyensya. Maayos na bukas para sa Pilipinas. Ating abutin ang pangarap ni Juan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan kung lahat magtutulungan. Tara na, sama-sama itaguyod ang siyensya. Maayos na bukas para sa Pilipinas.
Ang pangarap ko ay maging piloto.
Pangarap kong magkaroon ng Chief Alternative for Personal Hygiene.
Pangarap kong magkaroon ng mabilis at murang transportasyon para sa lahat.
Pangarap kong masagot ang malnutrition.
Pangarap ko pong magkaroon ng effective communication means for emergency.
Pangarap kong ma-maximize yung renewable energy source and to reduce the carbon dioxide emission.
Pangarap ko pong maging scientist.
Ngayon na o sabay akyat. Hawak kamay, tayo'y aangat, lilipad. Lipad.
A wonderful afternoon to our Zoom, Facebook, and YouTube participants. Welcome to the 15th installment of the Virology and Vaccine Institute of the Philippines, or the VIP's webinar series. I am Kimberly Neri from the Environment and Biotechnology Division of DOST ITDI, and I will be moderating this afternoon's learning session. We deeply apologize for the postponed webinar session yesterday and for any inconvenience that we may have caused. But here we are again, and we are very excited to share with you this newsworthy topic. SARS-CoV-2 variant Omicron is currently one of the leading topics in news and other public discussions because of its rapid rise in the number of cases around the globe. This afternoon, we will be enlightened by one of our Balik Scientists in the topic COVID-19 variant Omicron. This one-day webinar is part of the Virology and Vaccine Institute of the Philippines, or the VIP's information and education campaign on virology and other related topics. This learning session is made possible by the collaborative efforts of DOST Central Office, DOST ITDI, DOST PCHRD, DOST PICARD, and the DOST Balik Scientist program. With all the excitement, let us first check the number of participants here in our Zoom meeting, as well as our audiences from our Facebook and YouTube live streams. Currently, we have participants from Zoom. We have 31 participants. We have 33 participants from Facebook, and we have 69 participants in YouTube. Okay, so just a reminder, I would like to request our Zoom participants to kindly rename your Zoom account to the following format: affiliation _ full name. For myself, it will be DOST ITDI _ Kimberly Neri. This is done to be better facilitate the attendance as well as in the identification during the question and answer portion at the end of the session. I would also like to request participants to mute your microphones to avoid interruptions during the presentation proper. Thank you for your understanding. If you have any questions or clarifications for our speaker in this webinar, you may key it in our chat box in Zoom. Our audiences from Facebook and YouTube live stream may also type in their questions in the comment section so that our moderators can pick them up and be answered by our speaker. Lastly, to all our participants, regardless if you are in Zoom, Facebook, or YouTube, a certificate of participation will be given once you've answered the evaluation form, whose link will be given at the end of the lecture. So listen well and watch out for this. Now, before we formally start today's webinar, let me first share a couple of video presentations of the DOST VIP and the Balik Scientist program.
We are now living in a new normal. This COVID-19 pandemic marks as one of the global challenges experienced in this generation. It forces every sector of our society to innovate in order to move forward. We, at the Industrial Technology Development Institute of the Department of Science and Technology, is trying every possible ways to continue our service to our people, without compromising the safety of each and everyone.
recognizing the critical role of science and technology in economic development and progress. The Balik Scientist Act, or Republic Act 11035, was signed into law last June 2018 by President Rodrigo Roa Duterte.
This is actually putting into law a program that has been started by the Department of Science and Technology almost 40 years ago. But we need some legal support so that we can implement it in a better way. The Balik scientists will have an easier time in terms of coming here to the Philippines and rendering services.
Who is the Balik Scientist?
Experience and expertise of Filipinos who have made good practice of being scientists abroad, so that they can share whatever they have in terms of knowledge and wisdom to our own institutions, our own researchers.
My role and responsibility of a Balik scientist is to be not just a teacher or a facilitator, but also a pusher of innovations within the program.
Balik scientists are given support by the government for their stay in the country and are likewise provided with a wide array of benefits to ensure their maximum output.
The best incentives or privileges or benefits are having to be exposed with our farmers.
My greatest privilege would be doing collaborative work with fellow Filipinos.
The DOST, as the leading agency in charge of the Balik Scientist program, is tasked to facilitate the placement of the Balik Scientist among its priority areas from its sub-agencies. P-Card, PCHRD, P-Shared, Partnering with the DOST are the host institutions, private or public entities, providing the appropriate resources to the Balik scientist in the completion of their research activities and other tasks.
I think the role of the institution is to give the space or the laboratory needed for the program or the project.
Working together, the DOST, the Balik scientists, and the host institutions have proven the importance of collaboration and cooperation, critical of any nation's vision for success.
I am an advocate of Balik Scientist program. Okay, being one, I really truly felt that Balik Scientists would be able to help in uplifting the economic growth of the country.
My hopes is to really contribute to the space agency. It's very, very pragmatic, and we will need everybody's help, and also promulgate the STEM program here in the Philippines.
We really need to encourage other Balik scientists or other scientists abroad to give their time. They need to give back and help the country.
With the enactment of the Balik Scientist Act, the country is looking towards a stronger and more solid science and technology foundation, propelling the nation to further heights. Change has come indeed for science and technology. Science for change. Science for the people.
All right, science for the people. All right, that was our video about the VIP program as well as the briefer for the DOST Balik Scientist program. Thank you so much, DOST ITDI, for preparing the clips. Going back, last December 1, Dr. Vic Ilag, a Filipino Balik Scientist, had a webinar tackling the use of botanicals for antiviral therapeutics. During this webinar, he discussed the stages of the viral infection and identified targets for intervention. A good example of this are the botanical products that may have possible therapeutic effect against COVID-19. In addition, he also discussed traditional drug discovery pathway and the development of botanical products. I know that you are now excited to listen to our topic this afternoon. But before that, allow me to introduce our speaker. She is a doctor of veterinary medicine from the University of the Philippines, Los Baños, after graduating with the highest distinction. She then pursued her master's and doctorate degrees in molecular medicine science at Chonnam National University at South Korea. As a master's and PhD student, her studies were centered on the important viruses for global health concern. Following the receipt of her degrees up to the present, she has also been working as a postdoctoral fellow in Professor Ian Goodfellow's laboratory in the Department of Virology at the University of Cambridge in the United Kingdom. She is currently developing experimental systems investigating host-virus interactions of human norovirus and SARS-CoV-2. She also has been directly guiding PhD students, visiting scholars, laboratory technicians, and collaborating scientists. Her mentorships and collaborations had led to the publications of peer-reviewed papers and several manuscripts. Today, she has published over 40 articles in the international leading journals, including Science, Nature Magazine, and PLOS Pathogen. Finally, she volunteered to work at the coronavirus genomics team at Cambridge, playing an important role in the UK national response for COVID-19. Furthermore, she has also been part of the SARS-CoV-2 collaborative projects that involve in the establishment of serological tool and small interfering RNAs in activation assays. It is my pleasure to introduce and welcome a pride of our country, our very own DOST Balik Scientist, Dr. Myra T. Hosmillo, to talk about the COVID-19 variant, Omicron. A virtual round of applause for our speaker. Good afternoon po, Dr. Hosmillo. The virtual floor is now yours.
Thank you very much, Kim. Isang magandang hapon po sa inyong lahat at isang magandang umaga mula dito sa Cambridge. Unang-una sa lahat, I really have to say my apologies for not being able to make it yesterday. But today I'm here and hopefully I will be able to give you the updated information about Omicron. Okay, so let us begin. One very important topic recently is Omicron, and that was highlighted by Kim. Also, I want to highlight that there are a lot of variants that we have experienced worldwide. And the big lesson for that, and I have just picked this up from one of the presentations from Bill and Melinda Gates Foundation, is that variants prove that all health is global. So no country is safe unless everyone is safe. And I think it's one of the purpose why we try to keep educating people in different countries. And a very good example is why DOST and ITDI is making these efforts for us to be able to inform the Filipinos and the country, of course. Can you hear me clearly, by the way? Kim, can everyone hear me clearly?
Hello, Doc. Yes po, you are audible.
Okay, thank you. I just worry because I've given a few presentations recently, and after finishing the whole presentation, there is either my presentation or have a problem, or the way I speak have a problem. So I think before I start, I'd rather have that I'd rather make sure that everybody can hear me very clearly. Okay, so let us begin. So Merry Christmas to everybody. And we're really ending the 2021. And on an unfortunate case, we are again beginning a sense of uncertainty. And that is because of the emergence of Omicron. But before that, I would like to let you know as well that there are a lot of extraordinary achievements during the year. And one of that is at the beginning of 2021, we have several vaccines that have really shown good effectivity in randomized trials. As of the moment, there are now 27 authorized vaccines, and there are another 200 candidates that are in development. And this is a massive achievement because years ago, we would not be able to develop drugs or vaccines for over 10 to 15 years. And for us to be able to have this available and have it useful while we combat the different variants is really important. Secondly, we've got novel treatments, which also provided further options to prevent death and disability. So currently, there are a long list of previous drugs that are being trialed for COVID, as well as new drugs that are being established. I also am part of a collaboration where we are testing siRNAs to be able to, and we are testing siRNAs against live coronavirus infections. There is definitely a massive vaccine rollout programs that has taken in many countries. For example, in Portugal, there is 88% vaccine rate. In Chile, there is 86%. In Cuba, it's 83%. In Singapore, there is 83%. Although our countries is really a little bit lower, I think there is an encouraging increasing percentages of vaccine rollouts back home in the Philippines. However, there is a clear political failures to cooperate and find solutions at the global level. It is just sad that still there are very rich countries just trying to who have pledged to donate vaccine doses, but still until now they are unable to deliver. And there are still problems in relaxing intellectual properties agreement, especially in terms of sharing reagents or sharing vaccines, which will be really important to better understand coronaviruses or SARS-CoV-2. And I think the highlight of the ending of our year is really unprecedented, and unprecedented. And this is the emergence of the Omicron. And it threatens a new setback and further compounding harms of the pandemic. But I assure you that science and a lot of scientists around the world has been immediately studying this, and rigorously, intensively studying this. Over the year from 2020 to 2021, we have gained a lot of knowledge about the molecular virology of SARS-CoV-2 and the effects of various mutations that are seen in previous mutations as well. This means that whenever we have got new variants, we better understand the constellations of mutations in the genome sequences, and we can make a much better educated predictions. But I also want to note that even we have a lot of knowledge about the coronaviruses or the SARS-CoV-2, still there are a lot of unknowns, and there are actually a lot of unexpected findings. Today, what I really want to aim is to help you understand the concept of virus evolution, and also to give information about the current knowledge on SARS-CoV-2 B11529, or GRA, if it's a GSAID name nomenclature, or 21K or 21L, which is a next strain nomenclature. But we all know that it is called by the WHO as Omicron variant. Just to give you a little bit of background, I know everybody knows that at some point in 2019, a new coronavirus jumped species from animals into humans, and these are probably horseshoe bats. And the epidemic the pandemic started in Hubei, China. There are suggestions that from bats, they have transferred in an intermediate host, and from intermediate host, it has transferred to humans. But these days, it has just become irrelevant just because there is a significant human-to-human transmission that we all experience. And so one of the major advantage for actually having a sustained human-to-human transmission is because it can use a common human upper respiratory tract receptor, which is called angiotensin converting enzyme 2. These are located primarily in the mucus and the epithelial cells of your nose, in your eyes, and all the way down to your respiratory tract. So the initial spread happened in China in December, only in one province. And immediately from December to February, you can see here the highlighted regions of the country and how that it really spread so quickly. And in early 2020, from China, there was a pause, and there are spillage cases from international transmission. And it was us in 2020 with at least 3 different variants. And this is us now with several other variants. And it's really, it is really crazy that everyone thinks that we will be able to stop the infection in the middle of the year. We all want to end SARS-CoV-2, but sadly, I think that is not the answer at this point. So it has just begun very widely, and the WHO has declared the pandemic in March 11, 2020. And there is a significant transmission all over the world, especially in Europe, and then in the USA. And then from this to big spillage, it just continuously to spread in almost every continent other than Antarctica. And what are we facing now? As of December 18, 2021, worldwide, we have 274 million cases with 5.3 million deaths. And the Philippines recorded 2.84 million cases with 50,000 deaths. And right now, we are probably facing the 4th wave, and it hasn't stopped. So I just labeled here that we've got the alpha variant, which is the UK Kent Beta, which is a South African variant, which what we call B1315. P1, which is a gamma variant, which came from Brazil. The very famous Delta variant, which started in India. And now the Omicron variant, which actually first reported in Botswana in South Africa. Before I go through specifically on Omicron, I just would like to show you that this is a genome organization of SARS-CoV-2 or COVID. And these are the compositions of the virus. And as you can see here, this is just the levels of changes that are seen in different parts of the genome. So as you can see here, in the first part of what we call the open reading frame 1, there are few regions which had shown an increased mutation. And spike, as we would expect, contains a lot of genetic mutations. And that is because that is the one that is being used by SARS-CoV-2 to be able to infect. And what this tells us is, if ever, so whenever we try to diagnose a virus, we try to diagnose using primer sets that are conserved, meaning primer sets that won't change so that we will be able to detect viruses over time. However, in the case of SARS-CoV-2 and with a lot of RNA viruses, because of mutations, it is possible to affect the ability to diagnose the virus. And a very good example for that is how right now, the way we diagnose or we do a preliminary diagnosis for Omicron is through a failure of diagnosis. So one of the primers targets spike gene. And then in the spike gene, there is a region that is deleted in the case of the Omicron variant. And so the detection of the spike gene gets failed. So if we have a gene failure target, that becomes a candidate for a possibility of becoming or looking further, because it is possible that it could be an Omicron variant. So I just want you to be able to observe that there are actually mutations around the whole genomic sequence, and there are different levels of mutations, and these levels of mutations affect the diagnostics. These mutations also dictates the nomenclature of how we call the new variants. And as of now, the clade nomenclature that is being used by scientists is either given by Gisade, next strain, and Pango lineages. They all have like different ways of naming viruses. But the WHO has formed a group called Virus Evolution Working Group, or TAG on, I think it's Technical Advisory Group on Virus Evolution. The names that discusses on what or how are we going to name the new variant and the specific variant. In the UK, the government was able to make a consortium of scientists, and that includes us, to be able to study the important or variants that are of interest, and to be able to study them from genotype to phenotype and understand their ability to protect patients, understand the replication cycle, and basically to better understand the virus biology. Early of, I think this is in March when the consortium was grouped, I already received a report, and that includes change mutation that could possibly be big in the Philippines, and that is called theta. And so right now we what we know are alpha, beta, delta, Omicron, and gamma, but actually there are a lot of mutations that are happening constantly. So here, this is just based on a pangolin lineage. These are all how they are designated, and this is how the WHO designated. And this is where they were found. And so this is just scientists that makes the plasmids for us. And this is the designation, whether it's a variant of concern or a variant of interest. And this is what we looked at, to look at the different mutations in a particular variant and look whether these mutations would affect the ability of the antibodies to protect against reinfection, or the ability of the virus to replicate better in a host, or the ability of the virus to get transmitted better. So we have been monitoring quite a few lineages, or a few strains. And I would like you to give a definition of a variant of interest. So right now, the WHO have defined the strains, or grouped the strains, or categorized them as to a variant of interest and a variant of concern. So a variant of interest is a SARS-CoV-2 variant with genetic changes that are predicted to affect virus characteristics such as transmissibility, disease severity, immune escape, diagnostic, and therapeutic escape. And it has been identified as a cause of significant community transmission, or multiple COVID-19 clusters. And it suggests an emerging risk to global public health. Right now, what is listed as a variants of interest are lambda and mu. So lambda originated from Peru, while mu originated from Colombia. So when they arrived, so we identify the mutations of this variants and identify whether this variant has mutation that may affect the virus characteristics. And then what we do is we test them immediately whether they are of concern. At the same time, these strains or variants are being monitored whether the number of cases are getting a significantly increase, and that which means it will answer changes in transmissibility. And the variant of concern is a SARS-CoV-2 that has been known to have increased transmissibility or detrimental change in COVID-19 epidemiology. It may or may already have shown increase in virulence and change in clinical disease presentation. A very good example for that is a Delta variant. So what we know for a wild type SARS-CoV-2 is that it affects respiratory cells. However, in the case of Delta variant, it has shown, as well as respiratory lesions, it has also shown a possible intestinal pathology. So with the Omicron variant, we are also looking at whether, just like Delta variant, aside from causing respiratory problems, it is also causing intestinal pathologies. And a VOC is shown to decrease effectiveness even when public health and social measures are implemented, even when we've got diagnostics, vaccines, and therapeutics. So it is concerning because it will affect the healthcare system, And once it affects the hospitals and healthcare systems, then it will definitely cause an increase or significant levels of deaths and fatality. So currently, WHO designated VOCs are alpha, B117, and GISAID. So I think what you will hear in public will always be alpha and B117. However, I think it is important to note that when you're looking at clade nomenclature, it's also important to know that they can be named in other ways. And I have shown you that there are several mutations. So these are just the most important mutations in this particular variant. And this clearly has shown an effect in transmissibility, which has proved to have resulted in a rapid growth in the UK and internationally. Beta variant originated in South Africa, and this has this particular amino acid change. And we've got Delta variant, which have a lot of changes. Actually, I just want to highlight that all of these 4 variants have possibly around 10 or less than 10 mutations in the spike protein, which are biologically significant. However, with the emergence of the Omicron variant, it has shown that it has 30 or over 30 mutations in the spike region and 15 other mutations outside of the spike region. And these mutations, some of which can be really very concerning. So why is Omicron? So Omicron has been named by the WHO. It has another nomenclature, which is B11529. It's labeled by GISAID as GRA and labeled by NextStrain as 21K and 21L. It was first reported to WHO from South Africa and Botswana in November 24, 2021. But actually, the cases have been found in November 11 and November 14. The WHO has designated this variant in November 26 as a variant of concern. And as of now, this was yesterday, but actually as of today, there are 85 countries that have reported to have the Omicron variant. And this is just the whole picture of the Omicron variant around the world. But more importantly, the variant has a large number of mutations, and which some of those are really concerning. So this is a structural image of a spike protein. So the spike protein, this is a spike protein of a Delta variant. As you can see, there are important mutations. However, when you compare to the Omicron variant, and this is the RBD, which is the receptor binding domain, and this part, especially of the Omicron variant, has been shown to have several mutations. Some of those mutations, we sort of know what they're doing. Some of those mutations, we can guess, but there are a lot of other mutations that we totally don't know. So if you look at the whole genetic composition or sequence of the Omicron, there are not just changes. So when you see, for example, this is a spike, and this is an amino acid at position 67, which is changed to V. So that's how we read it. So that means that is a mutation. And there are, aside from mutation, there are also deletions. So there are deletion and mutation in from this part of the virus around the spike and at the other parts of other end of the virus. As I have said, there are amino acid substitutions that said to be convergent, meaning there are mutations that are very important, which we already know, which we have seen in other variants. And I will go through in all of that and the important ones in a while. So what are the likely effects of the Omicron? Early on, when the Omicron was detected and sequenced, and where the single nucleotide polymorphisms, what called SNPs, or changes, have been published, we know that there are overlapping mutations with alpha, beta, gamma, and delta, which are associated to impact of a PCR test. And that is called an S gene target failure. So when there is an S gene target failure, it is likely a new variant, but this time it's likely an Omicron variant. We know for a fact that, for example, N501Y is affecting the furin cleavage, and that affects allows the Omicron or the viruses to have a better binding affinity. There are changes that we know have affected transmissibility, and also, for example, N679K and E484K. These are mutations that have been shown to partially escape antibodies. So some mutations are overlapping with the previous other variants. Some mutations not in VOCs, but we sort of know what they do, and these are mutations that we know help virus to become more infectious or make it harder for antibodies to attach or kill the virus. And these are the other list of mutations that are largely unknown. And so we're looking at what their roles are. I can, if you want me to go through various changes of mutations in details of different variants that have been shown to be affecting the transmissibility, and for the others, I can go through with them later. But what we what scientists also look at is the changes of this mutation in relation to the structure of the protein. So this is the virus spike. The virus spike has the receptor binding domain, which binds to the receptor called ACE2, or angiotensin converting enzyme 2. The spike also have a fusion protein. This is located here. And then it is required to expose, and the host protein, or our cells, human cells, have proteases that clips it and allows the virus to enter its RNA into the cell. So you can see here, these mutations, this has just been reported in Twitter, but you can see here that you can see here that these are mutations that change characteristics of the Omicron. For example, if we look at number 1, the combination of mutations of K417N, 477, and N15 is thought to be affecting antibody evasion strategy. Deletions in position 69 and 70 mean the variant can be detected with some PCR test without the need for full genomic sequence. But I just want to note that in the case of Omicron, there are at least 3 sublineages, BA1, 2, and 3, and 2 of those have deletions in 69 and 70, but one of which doesn't have. So generally, when you have spike gene failure, you will be able to detect Omicron. But it is also possible that if you don't have that failure, you will still have Omicron, because there are some other sublineages of the Omicron that do not have the 6970 deletion. There are 4 new mutations that are identified as Q339D, 371L, that may create additional obstacles for certain antibodies. These mutations here around the furin cleavage site can be associated to transmissibility. The deletion mutations, not part of the spike, has been thought to aid in immune evasion, and there are 15 mutations in the receptor binding domain, and this allows the spike to immediately or fastly or easily attach the virus to the cells itself. So yeah, it seems like Omicron variant has come with a cloak of a much fitter and a greater advantage as compared to alpha, beta, gamma, and delta. So why is this important? Actually, variants have changed the COVID-19 endgame. In particular, it has actually shown with the appearance of particular variant, just for example, in South Africa, when the B315 was detected, it caused a surge of infections. It is somewhat the same in Brazil when the gamma was detected. It caused the country a surge in gamma infection. And what was very significant was in India, when an Indian variant strain was detected. Immediately after that, it caused that surge of infection in the country. And because Omicron was just reported like weeks ago, it's really very hard to know first what is its infection rate, what is its its transmissibility. So what scientists have done is really to look at the country of origin. And so this is how Omicron is in Africa. So before that, there is a lot of like recurrent surges of virus infection, of COVID-19 infections. And this surges were just due to South African variant, and possibly, I think it's mainly due to South African variant, which is beta. And as you can see here in December 18, there is a surge, and this time it's due to the Omicron variant. And if you can see the height of infection, it is highest among all the other previous infections. So the country has definitely experienced 3 previous waves driven by the regional variants, beta variant, and the delta variant. And right now, it's rapidly building an Omicron surge. And the recent report from the WHO that I've just seen in the morning was actually in South Africa, there is over 80% of their cases is already Omicron. And from 1% it has grown to over 80% of the cases. The Omicron in other parts of the world is being detected massively in the UK. Right now, we have over 11,000 cases of Omicron. And in London, there is 78% of Omicron, and in the whole country, there is 46%. The doubling time is now reported to be 1.5 to 3 days. And the risk of infection against Omicron is seemed to be 5 times higher than in Delta. However, the severity of infection is not really very, very clear. In Denmark, there is a surge of infection as well. There is a steep rise from November 28, and currently there's 785 cases reported. In Norway, due to an outbreak from a Christmas party, about 80 people got positive, and most of them have shown to be positive in Omicron. However, generally around the world, we still have Delta variant that is still predominant. In the US, for example, Delta variant is still dominant, and until December 17, there are a zero number of Omicron cases reported. So why is the surge in Africa? First, there are a number of new daily cases has been doubling in Africa much faster as compared to the country's 3 previous waves, as I've shown you. And the problem has been seen on reinfection. So although there are people who are vaccinated or infected previously, they seem to be reinfected. So that is a problem. The proportion of tests returning positive is accelerating. And this poses a risk on the ability of the protection or the changes of the virus to escape immunity or immune evasion. Also, we have to know that in Africa, they have just, I think, 11% of vaccinated, percentage of vaccinated in the population. So that may also be affected. So from November 30 in Africa, there are 44,373 cases. And in December 3, there are 16,000 reported cases. And as I've said, there's a very low vaccination rate. There is an anecdotal report that in South African hospital, a lot of young people have been showing the positivity in infection. But also, the country has a prevalence of 19% of HIV infection. And because these patients do not have a good or immunocompromised, and so it is possible that the virus lingers in these patients for a long time, which causes it to change over time. And these findings actually are not new, because early of 2020, we also had a report from our hospital where we monitored immunocompromised patients which were treated with convalescent plasma. And the virus has stayed for months and months and has showed various changes in its genetic composition. So it is possible. It is completely possible. And it's also possible that South African patients are quite different, and there is something intrinsically different from them. Whilst we talk on transmission, I would like to introduce or remind you again about the basic reproduction number. So basic reproduction number is numbers or a measure that is not affected. Is a measure of spread without any intervention. So the wild type Wuhan has a 2.5 RNA, Delta has 7, and Omicron has 10. However, with intervention in a real-time situation, and I think we now have what we call an effective reproductive number. And I think it is important to look at effective reproductive number rather than a basic reproductive number. And for Omicron, it's about 3.7 effective reproductive number, at least in London. So there are, because of this reproductive number, because we know how the virus evolved, because we know that there are mutations that will affect transmission, we know what is happening. Scientists from Europe are able to calculate or do mathematical predictions how that possibly there will be a surge in Europe in beginning of 2022, and also the surge in Africa could be a lot higher. So there are predictions, and I think the take-home message is that we are not ending COVID-19 yet. It will stay with us possibly, and it might stay with us just like how flu stay with us, and we will have to manufacture vaccines that will be appropriate for new variants. And actually, some vaccine companies and Pfizer, for example, have already designed this updated vaccines, and they are already in human trials. So there are a few questions. Are current diagnostic tests effective in detecting Omicron? The answer is yes. So also, because we can now, we know we can now detect a spike failure gene target, this sort of helps us to eliminate a sample to be possibly a new variant and to be possibly an Omicron variant. There are tests though that uses N as a target genes, and as of now, even with the mutations that are found in Omicron, this has not affected the ability, the effectivity of the diagnostic tests. However, it is always important that reading diagnostic tests should be in correlation with the clinical findings. Is the Omicron clinical presentation worse than predecessors? In Africa, they have reported that there is increase in hospitalization. In the UK as well, we have seen an increase in hospitalizations. We have recently had one case that has death due to Omicron. However, it doesn't really answer whether it's worse or whether it's caused more severe disease presentation. As of now, we still need to get more information about that. Are current treatments still effective against Omicron? So there are no evidence to show that treatments using these drugs or steroids, oxygen proning, all of these have been ineffective. They are still seen to be effective. However, the effectiveness of treating with monoclonal antibodies here is as an assessment. Recently, I have read that it has been reduced. And there are potential oral treatments that can be used. And recently, molnupiravir, which is also a drug that is targeting the virus polymerase, can be given orally and has already been shown to be effective. I don't know what other countries have approved this for use in patients, but it's now been approved at least in the UK. So are current public health interventions effective against Omicron? Certainly. So we should still wear masks, we should observe social distancing, improve ventilation in indoor spaces, avoid crowded spaces, and wash hands. That is still, I think, one of the most effective way to combat respiratory infections and respiratory viruses. Do immune responses from previous natural infection event remain effective in neutralizing Omicron? So I have looked at different vaccines and whether they still show protection or neutralizing titers against Omicron. There is so I will just show you. This is my data here in the UK. There is, and this is true for Johnson & Johnson, Moderna, and these vaccines, it has been shown to have reduced protection as compared to wild type. These are patients that are vaccinated with Pfizer with 2 doses. So there is a somewhat reduction in protecting each one of us from getting the infection. However, in protecting each one of us from getting severe disease, vaccines and all vaccines is still very effective. So what we do in the lab is try to check the neutralizing titer against the emerging variants, in particular, the variants of concern. So in other parts of the world, there are studies that vaccines are still 96% effective. Actually, for Pfizer, it's 88% effective, which is still very good. And some other vaccines have also at least 83%. The severity has not changed though. So updates on the Omicron biology. We know that we have checked that a few of the scientists in the consortium have checked Omicron replication in cell culture, and they have shown that Omicron tends to infect at an earlier stage as compared to the other variants. The replicant kinetics, though, how it stays is still the same as compared to all the other variants. Importantly, it has not evolved to use an alternative protein receptor as used by other coronaviruses. So it still uses ACE2. And it does not cause more severe diseases. It is not shown to cause more severe diseases in laboratory animals. So what should we do now? First, we do not panic and we don't need to overreact. We have dealt with variants before, including variants with which showed immune escape. So we certainly know what to do, and we have vaccines already. We sort of have armaments while we're battling Omicron. Maybe closing borders is no significant benefit, just because the virus are already in the country. And as you can see, it transmits very effectively. And so I think it's just impossible that you can stop it. Because there is higher transmissibility, that we get significant number of cases. The problem is we increase pressure on hospitals. And this is true even if the Omicron is leading to a worse clinical presentation. Definitely public health measures work, so we have to use them. And there's likely reinfections from vaccinated patients. So we just really have to be very careful. I just want to, this is one of my last slides, and just highlighting that this is Alpha variant or a Kent variant or the UK variant, which started early on in later 2020, and that it increased for some time, and it has got extinct as well, and it's reduced. So what the key message is that mutations will arise. Some mutations can be predominant over the others. It can replace the current mutations. Viruses will mutate because we are all different. We all have genetic makeup. We come from different backgrounds. Each countries have different ways of dealing with COVID, giving vaccines, the vaccination. There's several factors that have to be taken into consideration why we have mutations. But all of those mutations will always come to an end. There is always that's a classic epidemic curve. So they will come and go. And so the take-home message is that new variants will continue to emerge, but it's important to understand the phenotypes of emerging variants in terms of infectivity, transmissibility, virulence, and antigenicity. And that's what we're trying to do as scientists here. And I'm sure a lot of scientists around the world is trying to do as well. It is important to quantify the phenotypic impacts. We now know the genotypic changes. We want to know what impacts is it causing. Impacts of an individual mutation and impact of a mutation in combination with the others. So currently, authorized vaccines offer protection. However, it's important that you have to get boosted because we have data that has shown that booster, you will get better protections when you've got booster shots. And I'm talking about not just second booster, but also third booster, because the virus protection wanes over time and we're constantly exposed. And so especially the vulnerable ones have to get their booster shots. It is important that there's close monitoring of real-world vaccine effectiveness. We will have to continuously monitor emerging variants, and we will have to review evidence for boosters and next generation vaccines. And this all comes out with a changing landscape. Communicating promptly whilst we get new evidence is really crucial and it's really important. So yeah, with that, I hope I did not end with a grim ending for 2021. I genuinely hope everyone is healthy and we will all cope with the Omicron that is just trying to invade. With that, I would like to end my presentation and thank you everyone for staying with me and until today. And so I'm happy to answer questions.
All right, thank you, Dr. Hosmillo. Thank you for that very comprehensive lecture, Dr. Hosmillo. I'm sure our participants are enlightened and have gained a lot from this presentation. May I acknowledge the virtual presence of the Deputy Director for Research and Development, Dr. Christine Marie Montesa. Hello po, Dr. Montesa.
Hello, good afternoon.
Hello po, good afternoon. So now for the question and answer session, you can now type in your questions in the chat box for our Zoom participants or in the comment section for the Facebook and YouTube participants. Do we have here some questions in Zoom?
I think everything is clear.
As we wait for our participants to type in their questions, I would like to ask you, Dr. Hosmillo. So it was just announced that from the DOH that the booster shots interval are now shortened to 3 months for the second dose of a primary 2-dose vaccines, or at least 2 months after the primary single dose vaccine is given. Would this be as an effective measure for the reduction of the rates of the different COVID variants, or would I say to end this pandemic, or would it be possible to have a reinfection for those people that have had their primary 2-dose vaccines or primary single dose vaccines?
Yeah, so your question is whether it is effective to get vaccinated immediately after the first dose. And immediately means after 2 months, right?
Yes, Doc.
Okay. So first of all, with just one dose, we don't have protection against the new variants. And that is true even for Delta variant. And that's what we have now. With 2 vaccine doses, the protection we have is significantly lower as compared to a wild type. And so it is really important to get the vaccination. In our consortium, we have recently received a report where they looked at the waning protectivity following 3 weeks, I think, following vaccination. So what is clear is that 1 month following your booster shots, you've got good protection. However, there is no clear evidence of sustained protection. So I think, and I'm sure that it's effective to get a booster dose after 2 months. It is important. Our problem is, I think not so much of a problem people with good innate immunity, but we don't know who has very good innate immunity. I think this is a huge concern with people who are compromised. So because the virus is lingering around and it's just around everywhere, it's very easy to get infected. We're just exposed, completely exposed. And the only way to protect a compromised patient is really to get vaccinated. It is important to get that immediately after 2 months because there's evidences that are showing that it starts to wane in 2 months. But yeah.
All right, thank you po, Doc Hosmillo. Questions from the Zoom participants? Medyo nahihiya po sila.
I have just read here, while you are typing your question, I can give you more update from the WHO, which I just read in the morning. Yeah, so you remember early on in the pandemic, we always want to achieve an R0 of 1, okay? And I think the whole world has sort of achieved that. But with the Omicron, it has gone back to 2.56. I don't know exactly what we have in the Philippines, but if it's increased in one country in particular, so when the virus gets moved and it gets if there are spillage cases, then that will trigger an increase in a reproductive number as well. So yeah, I think that is significant.
All right, we have here from our Zoom participants, we have here Mr. Didley Susi from DOST ITDI. How do you see the evolution of the virus to be going in the next years? Will it follow the course of being a seasonal virus infection, much like influenza or flu, or the other way around? Is the virus evolving to a less virulent strain or far from it?
It's a very difficult question, to be honest. The virus evolved. The good thing about COVID, which is a lot better than influenza, is that it only have 2 mutations per month. And this is low compared to influenza. With 2 mutations, because it has a virus protein that is sort of corrects itself. So it has ability to produce lesser mutation as compared to influenza. Whether or not the mutation will be more virulent or less virulent is really very difficult to answer. What we're just trying to do at the moment is look at the changes that the virus do, and then following that, we look at how it affects the virulence. But I don't think there is a way where we can see that the virus would be, well, we can identify a specific change that would be more virulent. I have to stress though that the viruses, and every RNA viruses mutates continuously. So whilst you heard about Omicron, there is actually already over 100 times of changes that has happened in COVID-19. And so it's like 1 over, I don't know exactly, but it will be like 1 over 1,000 of a virulent strain over a non-virulent strain. So there will be some virulent strain, but the likelihood is still lower if we're looking at mutation changes. And also because the virus mutates so slow, I think the ability to change massively to become very virulent is also not very alarming. I also had experience where we had lambda strain, and so, or for example, just the theta strain in the Philippines. So, for example, the theta strain or the P3 was detected in the Philippines. We sort of know that there are specific mutations that could be nasty as the changes in spike. And so what we do is we monitor it immediately. But was it being monitored? The cases did not actually progress. It got extinct. So there would be maybe, it's really very hard to predict, but those situations happen. You may hear what is the next to Omicron, you may hear another one, it will go up and down. With the Kent variant, it went up, sustained a little bit and went down. It is possible that because we've got vaccinations, we've got good vaccinations, there is also drugs being given. It is possible that Omicron viral would just go up and go down immediately. It may not be sustained because as of now, hospitalization rate is not really significantly high. There is increase, but we don't know if it's just because there is increase in number of cases.
All right.
If ever people have questions online, and I will be able to, maybe if they are shy, they can email me as well.
Yes, you can email Dr. Hosmillo if you have questions. Okay, so in the interest of time, we will be ending our Q&A session and we will proceed to the presentation of the Certificate of Appreciation. And now to present the Certificate of Appreciation to our speaker, may I call on Engineer Reynaldo Esquera, the Chief of Environment and Biotechnology Division. Hello po.
Hi, team. Could you flash the, okay. Good morning, Doc Myra. Okay. So today I'll be awarding the Certificate of Appreciation, which states the Industrial Technology Development Institute. The Certificate of Appreciation is presented to Dr. Myra Hosmillo, Balik Scientist, University of Cambridge, for sharing her experience as resource person for the webinar entitled COVID-19 Variant Omicron, as part of the establishment of the Virology and Vaccine Institute of the Philippines program of the Department of Science and Technology, held online on December 21 and 22, 2021 via Zoom. given this 22nd day of December 2021, signed Annabel V. Briones, PhD, Director, DOST ITDI. Again, thank you, Doc Myra, and Merry Christmas.
Merry Christmas po.
Sorry, but I'm still on the road. And I see Doc Ted in the participants list. Doc Ted, also a Merry Christmas to you too. Thank you.
Hello po, Doc Ted. Yan. Merry Christmas po. And this time, may I request our participants to turn on your camera for the picture taking.
Are you going to count?
Yeah. All right. All right. So to formally end our webinar this afternoon, let us hear again from the Chief of the Environment and Biotechnology Division, Engineer Reynaldo Esguerra.
Okay. Well, I think this is. Do we still have a schedule for next week? I think this is the last of for this year, and really, it's really a pleasure to have Doc Myra as one of our resource persons for our Virology and Vaccine Institute program of the Philippines. And well, always I'd like to wish everyone a Merry Christmas and a Happy New Year. And I will see more of each other again for next year. And I do hope Doc Myra would be able to visit us very, very soon, because very recently we had one Balik Scientist who was able to come to the Philippines. So maybe in the very near future, next year, Doc Myra would be physically present.
I have flights going back home to the Philippines, so I'll let you know.
Okay, so again, good afternoon, everyone, and a joyous Merry Christmas and a prosperous New Year to everyone. Thank you.
Thank you, Sir Ray. Engineer Esguerra. Thank you very much to all who joined our learning session for today to continue our installment of the VIP webinar series in the year 2022. Here's the first and upcoming webinar entitled Molecular Virology, Genome Sequencing, and Bioinformatics by Dr. Christina Layson, to be held on January 10, 2022. So mark your calendars for the upcoming VIP webinar. And please answer this evaluation form flash on your screens to secure a copy of your certificate. This has been Kimberly Neri of DOST ITDI. Do like and follow the Environment and Biotechnology Division of ITDI Facebook page for the announcements regarding our future VIP webinars. Maraming salamat at maligayang Pasko sa inyong lahat.
Tumitilaok na ang manok, hudyat na ng pagpasok. Paglilingkod na walang kapalit, sa bayan ng aming hatid. Tara na, kaibigan, huwag kang magpaiwan. Gamitin ang dunong bansa'y susulong. Ating abutin ang pangarap ni Juan sa pamamagitan
hamon ay haharapin, mahirap man ay kakayanin.
Sa pinagsamang lakas at galing, tagumpay ay mararating.
Tara na kaibigan, huwag kang magpaiwan. Gamitin ang dunong bansa'y susulong. Ating abutin ang pangarap iwan, sa pamamagitan ng agham. Ang kaunlaran ay makakamtan, kung lahat magtutulungan. Tara na, sama-sama, itaguyod ang siyensya. Maayos na bukas para sa Pilipinas. Ating abutin ang pangarap iwan, sa pamamagitan ng agham. Ang kaunlaran ay makakamtan, kung lahat magtutulungan. Tara na, sama-sama, itaguyod ang siyensya. Maayos na bukas para sa Pilipinas.
Ang pangarap ko ay maging piloto.
Pangarap kong magkaroon ng chief alternative for personal hygiene.
Pangarap kong magkaroon ng mabilis at murang transportasyon para sa lahat.
Pangarap kong masagot ang malnutrition.
Pangarap ko pong magkaroon ng effective communication means for emergency.
Pangarap kong ma-maximize yung renewable energy source and to reduce the carbon dioxide emission.
Pangarap ko pong maging scientist.
Ngayon na o simulan na, humanda sabay